Good evening, or good morning, whatever it might be. Terry Kavanagh here. I'm here to present to you some work that we've been doing in the context of glutathione metabolism and e-cigarette vaping. So let me share my screen here and get going on the presentation. So today we're going to be talking about the susceptibility of to e-cigarette chemicals and the role of glutathione. This is work that was carried out in my laboratory, principally by Michael Anderson, with assistance from Diane, Colin, Savannah, Nikki, Lou, Zara, and Teo. So the outline for today's talk, introduction to glutathione, its relationship to e-cigs and vaping, the experimental design for our studies, the results, and summary and conclusions. First of all, what is glutathione? Well, it's glutathione is a endogenous tripeptide. That is, we make it in our own bodies, consisting of the three amino acids, glutamate, cysteine, and glycine. It's an important player in phase 2 metabolism, which involves the conjugation to and subsequent excretion of various endogenous substances and foreign chemicals. It also helps to maintain redox states within cells, and this is an important cellular antioxidant, which helps us to scavenge and detoxify reactive oxygen species and other oxygen radicals. Glutathione metabolism is a complicated process, but shown here in the lower portion of the screen is the glutathione redox cycle. This happens to be a very prominent reaction in our cells, where we're able to scavenge peroxides such as hydrogen peroxide with the help of glutathione peroxidase. And in so doing, our glutathione becomes oxidized to the disulfide GSSG. It's reduced back to glutathione by glutathione reductase, and ultimately the reducing equivalents for this come through glucose and the hexose monophosphate shunt. Glutathione is also synthesized in a two-step reaction indicated here. And it's a little bit more higher resolution and detail in this slide. Glutathione is synthesized in a two-step enzymatic process, again, involving glutamate cysteine ligase and glutathione synthetase. GCLC is the catalytic subunit for GCL, and GCLM is the modifier subunit. So GCLC has catalytic activity and is feedback inhibited by glutathione. GCLM lowers the Km for glutamate and increases the Ki for glutathione in that enzyme, making it much more efficient. We also use sometimes this drug BSO to inhibit the enzyme to test for the importance of GCL in various biological reactions. What's also important with this enzyme is the relatively common genetic polymorphisms in both GCLC and GCLM, which have been associated with predisposition to several diseases in humans, including cardiovascular, pulmonary, neurological, renal, hematological, immunological, as well as various cancers. Again, glutathione metabolism is involved in foreign chemical conjugation, and that's usually mediated by glutathionase transferases. So in summary, glutathione is a key player in the detoxification of many xenobiotic compounds, likely including many of the constituents of vaping liquids and subsequently generated aerosols. And where does this project fit in? Well, many studies of e-cigarette chemical constituents have shown there's an abundance of reactive chemical intermediates formed by the high temperature pyrolysis of the commonly used humectants and carrier solutions of propylene glycol and glycerol. Many of the flavoring agents present in e-cig liquids are themselves reactive carbonyl compounds. And these reactive chemicals and carbonyl compounds tend to be thiol reactive and are likely to be toxified by the thiol glutathione directly or through glutathionase transferase mediated conjugation reactions. Therefore, cells and tissues with different glutathione levels may be differentially sensitive to the adverse effects of these many reactive chemicals. If this is true, then this could have implications for people who may have different levels of glutathione in their cells and tissues based on genetics, diet, or other co-exposures. The e-cig compounds being studied in this series of experiments include formaldehyde and acetaldehyde, which are shown at the top there, the pyrolysis byproducts. And then cinnamaldehyde and diacetyl are flavoring agents commonly used in liquids. And all of them have these reactive oxygen carbonyls present in them, which means they probably react with glutathione. Again, the byproduct formation that occurs at the high temperatures of those heating elements in the e-cigarette devices is shown at the top. This is complicated chemistry, but suffice it to say that when all is said and done, the end products are these reactive aldehydes. So in our project, we decided to approach this by through a genetic approach, and to more thoroughly characterize the role of GCLM and glutathione synthesis and scavenging e-cig reactive chemicals. We utilized a GCLM mouse model that was previously developed in our lab, where the animals are deficient in GCLM. And through selective breeding of those mice, we created an immortalized liver cell line, as well as from these GCLM knockout mice, as well as from GCLM wild type normal mice. The immortalized GCLM null hepatocyte line, GCLM knockout cells, was about 10% of the normal glutathione levels present in the GCLM wild type cells. And we cultured these cells in vitro and exposed them to 4 e-cig chemicals listed there. We then assessed cell survival, activation of the GCLM gene promoter, glutathione depletion, and global changes in gene expression in response to these exposures. Now, that mouse that I talked about has a very interesting genetic complement, and that is that we replaced exon 1 of the GCLM gene with the beta-galactosidase neomycin phosphotransferase fusion gene. And this allows us to actually assess the promoter activity of the GCLM gene, which signals its activation, signals an antioxidant response. So if beta-galactosidase activity goes up in these cells, then it suggests that the GCLM promoter is turned on. If it goes down in these cells, it's either because it's been a toxic reaction or because the GCLM promoter has been turned off. This is kind of shown in this slide here. These are micrographs of liver taken from the wild type mice on the left and the GCLM null mice on the right. You can see when stained with this beta-galactosidase substrate called XGAL that the null mice show this strong positive reaction indicating beta-galactosidase activity and or a GCLM promoter activity in the various cells in that liver. So we took these 4 chemicals and exposed our cells to them and at various doses, and then assessed cell viability, GCLM promoter activity, glutathione depletion, and gene expression. So on to the results. Formaldehyde showed a classic dose response where if you increase the doses, you see decreased viability in the cells in both cell types. Interestingly, at those intermediate to low to intermediate doses, the GCLM knockout cells actually were showed to be slightly resistant. Now, this may be due to the upregulation of various protective enzymes, and I'm going to talk about that later in our gene transcription and mRNA-seq data. They did show an enhancement of the GCLM promoter activity at the lower doses. That's in support of that contention. And also, we did show that the higher doses of, that the intermediate doses, of formaldehyde actually cause an induction of glutathione biosynthesis relative to the controls. And this is in contrast to the positive control here, which is a compound that is known to deplete glutathione called hydroquinone. We also showed a decrease in the effects of formaldehyde on the wild type cells at the higher dose, again, reversing that induction. So acetaldehyde, in contrast, showed very little effect on cell viability. In fact, there's an increase in cell viability at the lower doses, lower to higher doses of acetaldehyde. This may be because acetaldehyde is metabolized in liver to acetate, which is a cellular energy substrate. and very slight modification in the GCL promoter caused by acetaldehyde, but it did cause some depletion of glutathione at the intermediate to higher doses in the wild type cells. And this was probably because of, again, its reactivity towards the thiol group of glutathione. On the other hand, there was no effect on cell viability at these doses. Cinnamaldehyde showed very interesting differential response between the wild type and knockout cells, the knockout cells being quite sensitive, especially at those intermediate doses. And the higher doses did seem to affect the level of survival in the wild types as well. Interestingly, in the knockout cells, we did see at least one dose at 100 micromolar that did show a significant induction. But the higher doses, probably because of the increased lethality of cinnamaldehyde towards the knockout cells, showed a decreased response in that beta-galactosidase activity. And this was complemented by glutathione levels being depleted in the wild type cells at those intermediate and somewhat higher doses. Diacetyl, another flavoring agent, didn't have much effect on cell viability until the highest levels of exposure in the wild type cells. The knockouts were not significantly effective even at that higher dose. We did see an induction overall in the in the GCM promoter activity, although in individual doses this was not statistically significant. It did cause some depletion of glutathione in the wild type cells at the intermediate doses, but induction at the highest dose, again, suggesting a turn on of the antioxidant response at the higher doses for the wild type cells. Now, if we look at the pathway analysis from the RNA-seq data, we can see, first of all, through principal components analysis, the biggest difference between these two cell lines, the biggest difference we see is the two cell lines themselves. The X's are the knockout cells and the circles are the wild type cells, showing that the amount of variability in the data is mostly attributed to this genotypic difference between the two cell types. And if we look at the differential gene expression of these cells as they are exposed to the different chemicals, the cinnamaldehyde and the diacetyl, we can see that there are a large number of gene expression changes. However, most of the gene expression changes that are present, excuse me, for some of these exposures is due to the fact that the wild type and knockout cells already have basal differences that are so different, as indicated in line 8 there. Now, what we did is we removed from the further analysis those exposures that didn't cause a significant number of gene changes, and with the remaining data, we submitted them to our pathway analysis, and showing here the effects of cinnamaldehyde at 100 micromolar in the wild type cells, a dramatic upregulation of a number of genes involved in drug metabolism, especially those that are P450 and other enzymes, phase 2 enzymes. Glutathione metabolism, as expected, is upregulated. And if you look at the genes that are listed there, the various glutathionase transferases seem to be some that are most affected, as well as the UGTs, which are also another set of phase 2 genes. And this is an important result because it shows, in fact, that cinnamaldehyde is inducing a number of these protective metabolism genes in the wild type cells. And the knockout cells, diacetyl actually had a very interesting effect on focal adhesion and extracellular matrix receptor interaction. This is an important outcome because it suggests that the ability of cells to communicate with each other through focal adhesion, plaques, as well as adhesion to the extracellular matrix present in the tissue, signals basically that these cells are having probably a difficult time with intercellular communication. The diacetyl effect on wild type is shown here. It's a mixed response with both upregulated and downregulated genes. Metabolic pathways were dramatically influenced, as well as the extracellular matrix receptor interactions here at this higher dose for the wild type cells. The knockout cells showed an interesting, again, effect on extracellular matrix interaction, as well as, again, at these higher doses, finally effects on metabolic pathways and antioxidant responses. So the summary of these results: formaldehyde exhibited relatively straightforward dose-response effect, with the GCM knockout cells appearing slightly more resistant at mid-range doses. This could possibly be due to the activation of some compensatory pathways, detoxification pathways. At high doses, formaldehyde appears to saturate, overwhelm the glutathione antioxidant system, which could cause issues in confronting other toxic insults. Acetaldehyde exhibited few effects in either cell line across all experiments and actually resulted in increased viability and antioxidant response. This may be due to the role of acetate, a compound derived from acetaldehyde metabolism, in various aspects of cellular metabolism and energy requirements of the cells. That being said, it is unknown if this may be due to the fact that the model system is liver. It is entirely possible that because of the liver's high metabolic rate, most of the acetaldehyde is converted to acetate before being able to exert any toxic effects or some toxic effects. Given all the data, it seems unlikely that glutathione status plays a major role in modulating any effects of acetaldehyde, at least in the liver. Cinnamaldehyde had drastically adverse effects on GCM null cells deficient in glutathione, even at mid to low doses. The wild-type cells were more resistant to cinnamaldehyde's negative effects. However, their antioxidant response systems were saturated or overloaded at similar doses, and their glutathione content was also extremely depleted, which could make the cells more sensitive to secondary toxic exposures. There was little change in gene expression at the lowest dose tested at both cell lines. At 100 micromole, the wild-type cells upregulated a plethora of metabolic enzyme RNAs, presumably as an adaptive response to the exposure, which further confirms the picture painted by the other data. Diacetyl at first glance appeared to have very little effect on both cell lines until the highest dose. And surprisingly, there were no differences between the two cell lines. Like acetaldehyde, there appeared to be sustained activation of glutathione antioxidant system across all doses. And transcriptomic and pathway analysis revealed some of the underpinning molecular mechanisms of diacetyl's effects. Wild-type cells showed no gene expression changes at the mid-range dose. However, at this dose, the knockout cells showed marked changes, primarily impacting various aspects of cellular bonding and adhesion. And by the highest dose, both cell lines showed differential gene expression, where wild-type cells showed downregulation of mRNAs in the multiple different aspects of energy metabolism and biosynthesis, which could easily lead to inhibited growth and development. Knockout cells also showed downregulated mRNAs in metabolism, but also demonstrated downregulation of the mRNAs involved in multiple types of receptor interactions. So, in conclusion, collectively, these data show that formaldehyde, which is a toxic reactive carbonyl formed during pyrolysis, and two ESIG flavoring agents, cinnamaldehyde and diacetyl, are likely to cause adverse responses in tissues exposed to them with vaping. These studies were conducted in mouse hepatocyte liver cell lines with different glutathione synthesis capacity. We're now replicating this work in lung airway epithelial cells derived from the same GCLM null and wild-type mice. And these cells are more representative of the most directly affected target organ or cell type, that is the lung, of the vaping, and will provide a more translationally relevant data set. We also plan in the future to culture airway epithelial cells taken from people. These would be taken from leftover from tissue from lung transplants that have different GCLM and GCLC genetic polymorphisms to test the hypothesis that genetic, sex, or age-dependent differences in GCL activity and glutathione synthesis may also influence the response to ESIG compounds. If this is the case, ultimately we'll want to know if it translates to differential sensitivities of people to the adverse effects of ESIG vaping and to protect the most sensitive among us with appropriate risk communication and, if necessary, regulatory policies. So I want to thank my laboratory for doing this work. Michael Anderson conducted most of this work as a requirement for his MS thesis, and he was assisted by Diane, Savannah, Nikki, and Colin. And our NYU collaborators, Dr. Terry Gordon and Monkey Chen, were helpful in both suggesting the work as well as interpreting it. Edge Center Functional Genomics, Proteomics, and Metabolomics Core and the Biostatistics and Bioinformatics Unit helped us a lot with that gene expression data and pathway analysis. Dr. Teo Bamler, Dr. Lu Wang, and Zara Afshraf-Yinajad. And the funding for this work came from NIH grants, one from the Cancer Institute, the other one from the Edge Center. Thank you very much, and I will enjoy talking to you on Friday about this work.